The plan today is to just briefly go over the start of the visual system from the last lecture, and then we'll talk about sort of transitioning into the visual system and what maps look like, visual maps look like in the visual system. That's the main topic for today. So last time we talked about the cells in the retina. So we have, you have the photoreceptor cells in the retina, and then there's a bipolar cell, and then there's a ganglion cell. And then there's some extra cells in this layer that sort of do additional circuitry. And the light, we said the light comes in this way. Light comes in this way, and it hits the photoreceptor there. And then the pigment epithelium, which is the back of the optic cup. Remember, the optic cup is a little outpocketing from the brain. It sort of sticks out. It's got two layers, and the outer layer is the retina, and the inner layer is this thing called the pigment epithelium, right here. It's like a little non-reflective backing to your eye. So this is how the light gets in. It hits the photoreceptors, and then the first thing we mentioned was that what happens when you actually look at the circuitry is that you start off with an input, like a photoreceptor input. And this happens in all different sensory systems. What happens is that input is kind of turned into sort of multiple loaves and fishes of different copies of that input. And then what happens is you come up to a processing stage, like somewhere in the cortex, and those streams, so these guys, sometimes these guys are called streams of information. So there are multiple copies of the information in the photoreceptor, but somehow emphasizing different things. And so those streams come in, and then, people were hoping that maybe we could just sort of follow them through. Like, say we have a stream that's more sensitive to motion or rapid changes, and that the hope was maybe we could just follow that through, up. But what happens actually is everything gets mixed all up together. And then what happens is a higher level stream will emerge out of that. And we'll talk about several higher level streams like that, so if you look at any one point, you can sort of see, this guy is sort of more sensitive to complex motion. This guy doesn't care about motion. Maybe this one cares about color. So you see these multiple sort of copies of the original data. The original data is there's actually only one copy of the original data from the photoreceptors. And so in the retina, the main distinctions that we talked about is so you have, cones here, and then the cones, you've got red and green cones, and then you've got blue cones. So you have red and green, but then the first thing that immediately happens is you break these up into on and off. And what is on and off? On and off means so if you look at the cones themselves, the photoreceptors, they actually are inhibited in response to light. So when you shine a light on a photoreceptor, it actually gets inhibited. And what the on and off business is the off stays inhibited, so the off bipolar cells will also be inhibited when you turn the light on, but the on bipolar cells flip it. So the on bipolar cells will be excited when you turn the light on. So that was the first stream. So you break these things up into streams, and there's actually a blue on and off, a separate blue on and off pathway. But then last time we talked about X and Y cells, and that was transient. That's Y, and X is sustained. So those are cells that respond to sort of rapid changes in the signal versus cells that mainly respond to the amount of the signal. So if you've got a signal, like you turn the light on like that, there are several parts of that signal. One part is where the brightness is changing. And so you'll essentially end up with one pathway that just pays mainly attention to where the changes occur. and then another pathway that pays mainly attention to just how much light is on. And this was the X-like pathway, or the sustained pathway, and this guy was the Y-like pathway, or the transient pathway, because it's really giving you a bigger signal when something changes versus just the total amount of it. That's not something that's present. That distinction isn't present in the photoreceptors. So that was made by the retina. So the retina cooked this up, and so then you have to subdivide these things into X and Y. So you keep so you can see you're really sort of multiplying the information, making multiple copies of the information, and that's this kind of motif of how the brain deals with sensory information applies to the somatosensory system. It applies to the auditory system. It applies to even to the pain system. There's a similar distinction between transient and sustained in pain. So it's a very general sort of concept of how the brain sort of computes. So that was a quick and dirty summary of last time. Does that sort of bring up any questions about the retina, or the photoreceptors, or the X and Y business, transient and sustained? And we talked a little bit about lagged. That's yet another way that you can divide these. You can have some cells that introduce a little bit of a lag in there in order to do computations for trying to figure out what direction something is moving. So that was called lagged and non-lagged. Yeah, so you can see the system really likes to do this, like take one copy of the information and differentiate it, make multiple copies of the same information, emphasizing different things. So any questions about the retina? So that was just getting the data into the system, very first stage. All good so far? Okay, so now let's sort of go into the system, further into the system. And so now you've got to deal with the issue of the fact that the eyes are in the front. So if you go way back, when eyes were simpler, they tended to be on the side. Like if you look at a little slug with eyes, it wants to cover as much of the visual field as possible. So the right eye kind of looks at the right side, and the left eye looks at the left side. But what's happened in several different groups of animals is that the eyes have rotated around the front. It's not just primates. We're primates, and our eyes are obviously around the front, but something like an owl has separately evolved this kind of system or a hawk of rotating the eyes around the front. There's even animals like some frogs. They start off with the eyes on the side, and then they move them around the front when metamorphosis happens. So it can even happen within one animal. But once you do that, then you have the problem of how do we sort of deal with the two copies of the world? Because if your eyes are off to the side and they're not overlapping, then you don't have to sort of deal with the issue of having two copies of the same thing. And so part of the complexity, comes from the two copies business. But if we just sort of put the basic idea to try to remember how all this left-right business works. So the basic idea is, the left world, this is the simplest thing to keep in mind. So left world goes to right brain, and vice versa. And the reason, part of the complexity is the lens. So just keep that will keep you from getting lost. So the left world goes to the right right brain. The problem is that the lens flips things. So when something goes into the eye, the cornea, which is the main lens of the eye, actually does a left-right flip. And that's probably, that left-right flip is probably responsible for why we have the rule of Sereno, which is like when you go past the midbrain, everything is kind of flipped, because the midbrain is where the visual input really first comes in. And that's probably like back in the really deep early parts of evolution, that's what caused this kind of left-right flip to happen. It's the fact that the lens sort of left-right flips objects. So that's the basic idea. Sort of the left world goes to right brain. So now let's just draw a little picture of the visual field. So here's sort of looking out at the world. And here's two eyeballs. So like we said, the cornea is the main lens. So when you look in your eye, what is the lens? It's really the cornea, the outer surface of the eye. That's why when you put a contact on, you change the shape of the cornea, you change the shape of the lens. The lens, on the other hand, you'd think like that would be the main focusing thing. That's just a little touch-up. So lens is just a touch-up on focus. Almost all the focus is actually done by the cornea itself. And so in fact, you can take the lens out and the eyeball still works pretty well. Or if you're old like me, like 60, almost 67, you have a really hard lens, and that lens is no longer flexible. It's like a hard super ball, so you can't, it doesn't change much in focus. So that's the cornea. So the cornea does most of the focusing. Here's the nose right here. And then we can divide up the visual field into 4 parts. So there's a part of the visual field that you can only see with your left eye. So that one is called, here's the left monocular. So that's only visible with the left eye. Even if you get rid of the nose or look above or below your nose, there's a portion of the world that's only seen by one eye. And then there's a big portion of the world that's seen by both eyes. So here's the left binocular, and then here's the right binocular, binoc, and then here's the same thing on the right monocular. So now let's just sort of look at all hell breaks loose as we actually sort of look at what happens when it goes into the system. So, back in the day, the eyes were off to the side. It was easy. The right world just essentially went into the right eye, and then that went into the left brain. But now we've got 2 copies going on. And so if we take something here in the left monocular, let's just trace that into the eye. So it's going to go in, it's going to hit the cornea and then get refracted. And then it's going to go into the other cornea, hit the cornea, and get refracted. And you can see it's going to go to the right side of one eye, also called the temporal part. So you can identify the retina itself by saying, you've got the nasal retina and the temporal retina. So it's the two left and right halves of the retina. And so this left binocular field is going into the, let me use a different color for that. So it's going into the nasal retina on the left side, but then it's going into the temporal retina in the right eye. So those are the 2 equivalent copies of the world that you're seeing between the 2 eyes. So it's the left side of the world going into the right side of each eye because of the cornea flipping things. So now we look at what happens, like what's the next stage? And so the next stage is is basically something called the dorsal lateral geniculate nucleus. And if we just make sort of an overall picture, so here's the retina. So the retina actually has more projections than just the dorsolateral geniculate nucleus. And so one of the main projections is to the superior colliculus. And we'll talk about that in detail because that's really involved in eye movements and attention, and it's all hooked up to the cortex. So, originally the retina 100% of the cells in the retina went to the superior colliculus. But there's there's some other branches. There's these things called the basal optic nuclei. There's, basal optic. There's something that's just in front of the superior colliculus called the pretectum. That's yet another pretectum. So the retina goes to that as well. It goes to the hypothalamus. There's, your day-night cycle needs to know, like, is it day or night? What should we do? Should we digest? Should we sleep? But then finally the one we're going to talk about here, which is one of the main ones, which goes to the dorsal lateral geniculate nucleus. So that's the dorsal lateral, because there's a ventral geniculate, that means knee, like genuflect, geniculate nucleus. So that's the one we're talking about. So that's the main relay nucleus. So now the plan is we go from these two retinas into the dorsolateral geniculate nucleus. And what does it look like? It looks like sort of like a really big taco, upside down. So and then next time, once we get all the audiovisual working again, I'll show you pictures of the geniculate. You can also look in the notes. I've got some nice actual micrographs of what the geniculate actually looks like in real life. So what is it? Well, it's kind of these very thick layers. And it turns out there's a lot of layers. So it's sort of bent like this into like an upside down taco. And then each of these layers is divided up into a contralateral and an ipsilateral. And all that means is something that receives an input from the opposite side, the opposite eye, versus something that receives an input from the eye on the same side. So this guy is the right dorsolateral geniculate nucleus. And so it's got these very thick layers, and if you look at what the projections are, you don't have to remember this, but we'll just write it down. So contralateral, so the top layer here gets input from contralateral, meaning like the right dorsolateral geniculate gets input from the left eye. And then there's an ipsilateral layer and another contralateral and another ipsilateral, and then an ipsilateral and a contralateral. So you can see it's sort of got a confusing thing. But these are all the different layers of the geniculate, but remember the geniculate is representing space. It's representing visual space. And so there's two directions to visual space. You can think of them like, how far are you from the center, or what the angle is, or you could do it in Cartesian coordinates, but there's two coordinates of a position in space. And so where is that? That's kind of spread out in this direction like this. So it's, like I said, it's like this very thick taco here. And so here's all the additional layers. And so the visual map of locations, different locations in space, is kind of spread out, in this direction across the surface of this, like that. So basically, those are the, these guys are the map coordinates. And so now the question is like, wow, what about all the layers? What are all these different layers doing? And so one of the things that the layers do is they separate these layers up here. Let's number the layers. So it starts off like 1, 2, 3, starting from the bottom, 4, 5, 6. So one of the things that's separated is the X and the Y business is separated. So these layer 1 and 2 mainly get input from the Y system. That's the more transient system. It's the larger cells. Larger cells are always more transient. But then there's also a segregation between the on and off business. So the Y on and off are not separated, but the X are, for better or for worse. So this layer 5 and 6 have mainly X on type cells, and then layer 3 and 4 have mainly X off cells. So let me just put down here. So C equals contralateral, just meaning just going from the opposite eye. And ipsilateral means coming from the same eye. Same side, yeah. Okay, so that's the geniculate. And so what does a point, what does a single point in the visual field look like in the geniculate? It actually, so imagine like we got this little point right here in the visual field. It's somewhere in that coordinate system of the map of the visual field. What that corresponds to is it corresponds to this little thin tube of cells that are going down through all the layers. And there'll be like an X-on version of it coming from the contralateral eye, and an X-off coming from the ipsilateral eye, and the Y. All those different things are kind of in this tiny little tube. If you go to some other part of the visual field, there'll be another little tiny tube going down through through all the layers like that. So you can see that the map, like if you just unfolded the taco, the size of the map is about the same size as the thickness of the map because of all this duplication of streams. So that, and then there's another geniculate on the other side. I didn't draw that in, but there's another one on this side, and that one is pretty much the same. And if you draw these basic connections here, you can see if we go back to this very beginning thing, basic idea, left world goes to right brain. So, left world, left eye is going to right brain. And so this guy, if we look at this contralateral connection, this guy's going to go and project, to the contralateral ones. So it'll go here, and then it'll go here, and it'll go here. And then what about the other side? So the other side is going to come out and start heading toward the opposite side of the brain until it gets to the optic chiasm. So this is the optic chiasm. And so last time, or 2 times ago, I talked about Siamese cats. Chiasm. Siamese cats have something screwed up with their melanin pathways. And surprisingly, just because of the way genes are so multifunctional, if you mess up the melanin pathways anywhere in the melanin pathway, there's some cells that are dying here containing melanin, and those are the ones that signal which side of the brain to go to. And so that gets screwed up in a Siamese cat. But if you're not a Siamese cat, once the axons get there, they'll see those dying cells with melanin, and they'll say, okay, we should stay on the same side. And so then they'll go down, and then they'll stay on the same side of the brain and project to those, to these other layers like that. So, and then of course, the same thing would happen. But when you look at this, you can say, there's a lot of left-right flips here. So that's why left world, goes to right brain. So left world goes to the right side of each eye, and then the right side of each eye goes to the right brain to line up the left and the right eye copies. So, is there any questions there about sort of a basic? So this thing is called a relay nucleus because not too much happens here. So there's no interaction between the different layers. There's interactions sort of within a layer, but there isn't really interactions between the layers. So it's kind of mainly sort of a relay nucleus. So that was the situation up until about 20 years ago. And then I always tell the same story. There's a little bit of a pecking order of people studying different kinds of animals. And so people who study primates that are more closely related to us, like macaque monkeys, like Asian or African monkeys, those people are a little higher in the pecking order than people who study smaller nocturnal primates. And so there are these little primates called bush babies, and the reason they're called bush babies is because they make these weird little noises that sound like a baby, actual baby. That's really why they're called that. And so people were studying bush babies, and they found, it looks like there's some extra layers. And they're not particularly, if you look in the slides, you can see they're quite visible there. There's like a whole nother layer right here of little cells, little tiny cells. And so those cells are called, the coniocellular layers. It turns out that's like the blue system. And there's one of these little thin layers in between the contralateral and ipsilateral, and there's another one in here. So there's actually 12 layers of the geniculate. So if we sort of divide it up, we have the X-like pathway, and then we have the Y-like pathway, and then we have this conio-like pathway, and that's basically blue. That's the blue system. And the Y system doesn't see color, so the Y system kind of messes up the color signal. It mixes it all together so that it can't see the difference between red and green. And then the X system is the red and the green system. So it's quite asymmetrical. It's just the result of how these things have evolved. So initially what happened is you had a distinction like in a squirrel, say, between blue and then one red-green system. And then what happened is in primates, the gene for the red-green pigment was duplicated, and so you ended up with two copies of it. Happens to be on the X chromosome. That's why men tend to be colorblind more often, because we only have one copy of it, and so that if that copy goes bad, well, then you only got one copy. So they discovered these additional layers in there in bush babies. Nobody paid attention because that's just bush babies. We don't care about that. And then somebody did exactly the same experiment in macaque monkeys. And that got published in Science magazine because it's kind of, macaques actually have them. So like I said, it's not like they're not visible. They've been there all the time. It's just that there's also a little bit of a bias in neuroscience against small cells. So like big cells, like Y cells, those guys are really good cells. And then like these little conio cells, well, they're kind of small, I don't know if we should pay much attention to them. And so for a long time, people just sort of didn't pay attention to them because they were smaller, but there's nothing wrong with them. They're carrying the blue signal. Okay, so that's our initial relay. So all this information from the retina, it's already been subdivided into multiple streams, comes down into the geniculate. So now, second half is now we're going to go up to the cortex. And so the next plan is this thing projects up to area V1 in the cortex. So And so now the plan is to kind of take a look at what this map looks like in the cortex. And if, I'm sort of like Mr. Map, and so I've done a lot of mapping of all different kinds of things. And there's large numbers of maps. So almost half of your brain, half of the neocortex consists of maps. So it's a major chunk. And so this is one of the first highest resolution maps in the cortex. And so we want to get some idea of what that map looks like. And so, and to talk about this, it turns out the map in the geniculate and in V1 is something that a mathematician would call approximately a conformal map. So this is conformal map. And so let's just talk about what that means. So a conformal map. So if you were taking a course in electricity and magnetism, you probably know you have like electric fields and magnetic fields, and they're at right angles to each other. You would do things with conformal maps, because conformal maps describe the way electric and magnetic fields are arrayed in space. Here we're using it for a completely different concept, but we just have to get some just vague idea what a conformal map is and how it relates to this business of where we split the retina into two halves, because it turns out that actually is a big advantage for making a conformal map if you split the retina into two halves. And there's kind of a functional reason for it, for how shape varies across the retina. So if we start off with a conformal map, a simple example of a conformal map. So we're just doing it in 2D. So we have a map with, like, say, x and y coordinates, and then we're mapping to another copy of that first map, but changing it somehow. And so if we make a little map here like this, so that's a little chunk of a little Cartesian map. And now we're going to make a conformal map. So the conformal map, it doesn't have to keep things the same size. So that's one of the first things. So here's our conformal map. So it doesn't have to keep things the same size. So that's an example of an approximately conformal map. And so what is, what's the definition of a conformal map? So if we were, the mathematical definition is a little tricky. Math always tries to reduce everything down to the minimum number of things. But the way that you can think about a conformal map is that it preserves local angles and it preserves local relative size. Local relative size. So the mathematical definition is more compact, but this gets across the concept better. And so what does the local angles business mean? Well, if we look at this map here and we just take one of these, say we just take this junction right here, you can see it's basically a right angle at that junction, and here's that same junction, and that's a right angle. Or if we look at this thing out here, you can see that guy's a right angle, and that would be equivalent to this junction. So the first part of a conformal map is that you keep the angles the same locally. So obviously over a longer distance it doesn't work, but locally it does. But then there's the second more subtle part is that you also preserve the relative size. So what does that mean? So that means, like, if we go out here and we look, you can see that, that map has a relatively small movement in the x and y direction. If we go out to this map copy, we can see we have a larger movement in the x and y direction, but the relative size between them is the same. So even though we stretched this part of the map, so I mean, if you think of this thing as just a rubber sheet, we stretched it into this shape here, and so obviously this part of the map is stretched. So this guy is stretched. So stretched, but same in both directions. That's the idea of a conformal map. So that's a conformal map. It preserves angles locally, but not at a long distance, and it preserves the relative size of things in different directions, but not at a long distance, because we obviously stretched this compared to when we compare back to this over here. So that guy got stretched. Okay, so there you go. That's a conformal map. You always wanted to know what a conformal map was. Impress your friends. So what the frick does this have to do with the visual system? So let's just take an example of the retina here. So say, I'll eventually erase that guy up there. So if we start off with the retina, one of the problems, if we take the whole retina, we've got a whole high density of cells in the middle of the retina like that. And then as you move out away from the center, the density of cells goes down. And so that's called the fovea, because that's why we can see really well with the very center. Like if you look at something directly on, you can see a lot of details. If I look off to the side and then pay attention to that same thing, wow, it's really quite blurry. You can barely read it. So you can't read way off to the side, for instance. So that's what the retina looks like. So if we just plot it across, if I just plot a density of the retinal cells, you would see there's this big spike right in the middle, something like that. If I just did one dimension of density. So this is, density. Now let's just take that map, and if we just take that map and we try to spread it out evenly to the geniculate. So that's the retina. The retina is all really dense, and let's just go take all the wires out of the retina and just try to spread them across as evenly as we can across the geniculate. What's going to happen? So it turns out there's going to be some problems. So the idea is we want to just take this retina over here and go to our dorsal lateral geniculate nucleus and just spread all these cells out evenly across it. Now, what is that equivalent to? Well, imagine this retina was like a rubber sheet with a big thing sticking up in the middle of it. That's kind of the problem we have. We have to take that rubber sheet and somehow stretch it so that it's all about the same stretchiness on the geniculate. And what is the problem? Well, the problem is going to be, let's just consider 3 points in the retina. So here's the center of the retina. So let's just go out a certain distance in the retina like that, and we'll consider, point, different color. Point B here. Here's point A and point B and point C. So those are 3 points that are next to each other before we've stretched anything out. So now we squashed that little sort of pointy little rubber sheet thing down and to try to sort of stretch everything into even distribution of all the parts in the retina. And what happens? Well, think about this. So it's really dense in the middle. It's really dense in the middle, and so that means if we're going to even this spike out, we're going to have to move these points away from the center a little bit because they're packed too closely in the center. And so if you move them apart a little bit, they're going to have to go out a little further. You have to go out a little further like that. And so they'll still be about the same distance apart because they're nearby points. So here's B and A. But now consider the other axis. So what's going to happen, unfortunately, is that when we look at point C, because we've moved out away from the center, point C is going to have to get further away from point B than point A is. So you can see here what's going to happen is it's not going to be a conformal map. So in other words, it's going to be stretched more in one direction than in the other, and that breaks this rule here that it preserves the relative local size. And so just the most straightforward way of trying to take a retina that has a high density in the middle and just squash it out to a uniform density of cells in the lateral geniculate is very hard to make a conformal map. And so what happens? Well, we cut it in half. And so if you imagine the retina, what does the retina look like? It looks like a half of an inside of a sphere. It's like a half a sphere. So if we cut it into two, it turns out then it's possible to actually make a much easier conformal map. And that turns out to be what the maps look like in the geniculate. And so we'll erase our actual sort of semi-accurate picture of the geniculate here, and we'll draw roughly what the map in the geniculate looks like. And it's hard to, when you think about how your visual system works, when you look around, nothing seems to change size. Yeah, question?
Was the conformal map the way we create like a hypothetical situation if the retina was a full circle?
Yeah. Okay. Yeah, that was the full circle case. Yeah, so now we'll illustrate the half circle case. But the thing that I wanted to just reemphasize, your visual system, you live in your visual system and everything looks about the same size. You look around the room. If I look at my finger, I see my finger. If I look at this finger, that finger didn't seem to change size. But what happened in real life? Well, I was looking at this finger and it was taking up like about a third of my V1 and geniculate. And then I looked over at this finger. Now this one's taking up about a third of the geniculate, and this one's taking up about 100th. So every single time you look around the room, everything is wildly changing size. Whatever you look at just blows way up. You look at somebody's face, you look right at their nose, they got a big nose. You look at their eye, now they got a big eye. You look at their ear, big ear, eye and nose got small. It's hard to imagine. That's actually the raw movie coming into your visual system. And it doesn't look like that. You just look around, everything sort of stays the same size. I look back and forth between those fingers, yeah, they look okay. Nothing. It's getting hundreds of times bigger when I look at this one versus looking at that one. It's kind of amazing. So what can we do if we just cut the visual field in two? So if we cut it in two, then we've got a simpler setup. And I'm going to just use these little symbols to indicate where we are in the visual field. So here what we're looking at is the visual field. So this guy is, looking out at the left visual field, so the left half of the visual field. So this is, this is the actual world, in which case, like fingers don't change size. And so the symbols we use thick dashes to indicate the horizontal line, and then circles to indicate the vertical line, and then we've got, upper and lower field. And then we can add a few more contour lines in here. Sort of 45-degree ones. Okay, so that's the visual field, but only half of a visual field, just the left half. So we cut it in two. So now it turns out it's much easier to make a conformal map. And so if we now map that into something that looks like this, so if we map the vertical line in the visual field almost to a circle. So that's the vertical line in the visual field, like that. And then here's the periphery. Periphery is very small because the density in the retina is very low out there. And then here's the horizontal line in the visual field. And then here's those other lines. So there's one of the other contour lines and another contour line there. And then here's our 45-degree angle line. You can see, it's bending everything around. But if you look at what's going on in the dorsolateral geniculate, like if you looked at the map in the dorsolateral geniculate, or V1, this is what it would look like. It would be a conformal map. And so what is the advantage of that? So we've done all this sort of math nonsense, but what's the advantage of it? Well, let's just take a simple example. Like say I'm just looking straight ahead at this point right here. And say, so let me just look right at that point. Okay, I'm looking at that point. Now say there's something, that I'm walking forward. So I'm walking forward and say there's a tree over here. So if there's a tree over there, so I'm looking straight at this point, and there's a tree. Now what happens as I walk past the tree? So two things are going to happen. The tree is going to move into my periphery, because as I get closer and closer to the tree, the angle of the tree is going to go out, so the tree will sort of move into my peripheral vision. But it's also going to get bigger because I'm getting closer to it. So in this particular special case, it's a common special case, walking straight forward. As everything passes you, as you get closer to it gets bigger, but it also moves into your periphery. And so those two cancel each other out. And so a conformal map is kind of a handy thing, because what happens is the tree stays about the same shape and size. It doesn't radically change its shape and size. You got closer to it, and so it actually got bigger on your retina, but it went out into the periphery of your retina, and then your conformal map shrunk it down. So one of the advantages of a conformal map is if you're trying to recognize objects by shape, so you're trying to recognize a face, and sometimes you look at the nose, sometimes you look at the eye, you look at various different parts of the face, and what you're trying to do is recognize it by shape. And these kinds of maps will preserve shape. They won't preserve size. So size can sort of go all over the place, but they will preserve shape. And so that's one of the, and if your bottom line is to figure out, is it this person or that person, you're interested in these subtle differences in the shape of the object. And so that's one of the possible reasons for a conformal map. I think I'll just continue with. I can't get into your. We can't get into the.
You can, but I can't. It needs your ID.
Why don't we just take? I'll slightly put off the cortical maps. Let's just take a look at the geniculate. Got to see some actual pictures. Any questions while I'm just getting this set up so far? good on the maps? So let's get in here. Oh yeah. One more time. Type cleanly. Okie doke. Okay, so I'll show you some pictures of the geniculate, but before I do that, I wanted to show just a few pictures of the previous. Okay. Alrighty. Okay, we've got some pictures. Okay, so this is okay. So this is a picture of the neural tube. So in the previous lectures, we talked about how the let me just zoom this guy a little bit. Okay, that's good enough. So we talked about the neural tube, and so this guy here is the neural tube rolling up. So it hasn't quite, rolled up, and these guys are the little neurons that are sort of crawling up and down on the guy wires. So that's actually the neural tube in the process of rolling up. So it really, it starts off, it just looks like a little tube. This is the skin here, right here. You can see that layer right there. That's the skin of the embryo. And so if you have like a spina bifida, what happens is the neural tube doesn't roll up, and so the actual nervous system is exposed to the air, which is not a good plan. So that's a picture of the neural tube. This is a picture of erase. There we go. This is a picture from the top of the same thing, and we mentioned there were these things called somites, which are those segments, kind of like your vertebrae of the body. And then this was a nice picture of just a little bit later in the neural tube. So here you can see the notochord, and remember the notochord came from the part of the gastrula, like where there's an in-pocketing, and the top layer above the in-pocketing, that's the neural plate that turns into the neural tube, and then the top layer of the in-pocketing itself, that turns into the spinal cord, into the bony part of the spinal cord. So that's the notochord there, but here's the neural tube. And we're at a slightly later stage, so this is the ventricle down here like that. And there's the whole neural tube. And so when we were talking about how those cells, they crawl up here, and then they turn around and crawl back down here, and then when they get back down right to the ventricle, they divide. And then just one of them will, crawl off into the sort of space off to the side to start building up the rest of the brain. And when you look at these pictures, it's just like, it doesn't look anything vaguely like a brain. But that's how these things start. They all start sort of very simple, sort of geometrically simple. And here you can see one of the somites off to the side. That's basically one of the segmented muscles of the back. If you're eating like a fish, like if you eat a salmon, a single somite is like one of those little pieces of. If you just pick apart one little piece of the muscle of the salmon, that will be one somite. Okay, so let's try to cleanly erase that and look at a few other pictures. So this guy, that was a picture of the eye. And here's another picture of about the same thing. So that's the optic cup. Now, it doesn't look like much there. here you can get a little better picture for what the optic cup looks like. So this, it sort of looks like what I was drawing. It's amazingly sort of generic. So this is the, oops, this guy is the retina right here. So the retina goes like this, and then down to about there, and then like that. So this part is the retina right here. So that guy's the retina, and then behind it, it goes around like that. Behind it is the pigment epithelium. So this thing, this thing over here is PE for pigment epithelium. So that's the backing of the eye that touches your photoreceptors. And late at night, it chews off the top of the photoreceptors to rejuvenate them for a new day of looking at bright objects. And then this thing is the lens. So you can see the lens is just a little, is basically just a little vesicle in there. And it initially forms by an in-pocketing. Here you can actually see the in-pocketing, where it's going down in like that. That's actually what forms the lens. So the eye actually does look like that. And so here's this this retinal axon will then just grow in toward the nervous system, and it will quickly figure out I've got to go to the other side, so if this is the right eye, I got to go to the left side. Okay, so what did I put that in there for? Yeah. Yeah, okay, that was about dogs, evolution of dogs. This is a very similar thing that happens in the formation of the ear. So the formation of the ear is also formed as a little in-pocketing. The cochlea initially starts off as a little in-pocketing. And the specification of the somites, we talked about the specification of the somites, turns out to be done by a set of genes called homeotic genes. And these genes are genes that kind of specify like what segment you're in. And these were originally discovered in flies. So if you mutate a fly, there's something called a mutation called antennapedia, which doesn't sound too good. And what that is that's the antenna right there. You're supposed to have an antenna sort of sticking out of your head if you're a fly. Okay, that's all good. And here is a sad fly that has a leg sticking out of where its antenna is. And so what happened there is that the specification of which segment you're on got screwed up. And so there's a high level set of genes that specify which segment is which. And then there are lower level genes that just make a leg. And so the lower level genes say, okay, well, I guess we're in the segment that's supposed to have a leg, so we'll make a leg. And so it will actually sort of generate a leg coming out of the antenna. So those genes were originally discovered in flies, but it turns out we've got them too. In fact, ours are a little more complicated, not that much more complicated. We have maybe like 2 times as many as a fly does. But those things specify the segments of the body, but not just the segments of the body, they specify the segments of the brain also, the front to back segments of the brain. So let's take a look at what the neural tube actually looks like. So I drew the diagram that I want you to reproduce, which is that diagram of the spinal cord and the medulla and the pons. One more time. I think I will finally get it this time. There we go. If you actually look at that diagram in real life, it's a little more folded up. So I drew a picture of it from the top. So the spinal cord is down here, and then this blue part is where the fourth ventricle is open, and then the fourth ventricle closes, and then you get the midbrain, the mesencephalon. And then there's another opening, the third ventricle, and then you get the 2 cerebral hemispheres as 2 outpocketings. So that's actually what it looks like sort of more in real life. if you look at this is the picture here where you can actually see so this is the opening of the fourth ventricle that I draw like a little, a little triangle. And then here it's closed back up again. The ventricle closes back up again there. And you can see the cerebellum sort of grows over the top. And if you look, this is the spinal cord, so the neural tube is closed again down there. Yeah, so it doesn't look, like I said, it doesn't look too much like a real brain. This is a picture of a frog brain, which is very similar to the diagram that I made, so that you can sort of get an idea. So you can see here's the opening of the fourth ventricle, and then it closes. There's the midbrain. That's the pons midbrain junction right there. And then here's the forebrain, which you can see is two little separate outpocketings for the left and the right, but it doesn't look like much compared to a primate. Okay, and in another lecture I talked about the subplate. And the subplate is a temporary set of cells that forms when the cerebral cortex is forming. And so if we look at let's look at the end, the end game here. So this is the cerebral cortex. So this is layer 1 up there, and this would be layer 4 over here, and layer 6 down here. And this is the white matter. So that's the end stage. But during the development of the cerebral cortex, and in particular during the development of maps in the cerebral cortex, there's this thing called the subplate. And the subplate is this gigantic thing. It goes all the way from here to here. So you can see it's much bigger. It's even thicker than the cortex. So this guy is the subplate. It's got all these layers in it. So that thing forms before any of the actual neurons in the actual cortex. This is the actual cortex here form. And they're not just sort of sitting there, they actually get connections. So the lateral geniculate, for instance, grows in and contacts these guys temporarily. And then what happens is, eventually, a lot of them die off, and other ones essentially migrate through each other up into what will eventually form the final cortex. And then this whole thing goes away. So you can see that whole subplate is basically sort of turns into the same space as the white matter. So there's this complicated development of this whole giant structure with a whole bunch of layers in it and everything that sort of just somehow temporarily stages making maps in the cortex, because you don't see maps in the cortex without something like this going on. So that's the subplate. Is the question? No? Okay, so there was a lecture on the subplate, if you're interested in that. Okay. And I talked a little bit I'm almost done. I talked a little bit about how do the folds in the cortex work? So, if you look at the cortex, you can see it's all folded up, it makes things, life hard. Turns out the way that this mainly works, only recently figured out, the way this mainly works is that the white matter acts like a hard sort of armature, and the cortex is like an expandable layer. And so where do folds come from in the cortex? What happens is the white matter, all the axons, act like a hard sort of armature that doesn't change shape. And then what happens is the cortex is a layer on top of that expands relative to the armature. And so as it expands, what will happen is it will kind of get thrown up into folds because it's sort of stuck to this hard white matter that's not that's not expanding. And so that's actually where most of the folds in the cortex come from. They come from sort of as the gray matter with all the neurons in it puffs up, that gray matter will kind of throw itself up into folds. And so people have started sort of making models. This is an actual physical model of it where you make like a little gel, a hard gel sort of for the armature, and then you make a softer gel that plumps up, and you can see many of the standard places that the brain folds will appear. Okay, and last but not least, let's go down. Oh, I gotta look at this for a second. So any guess what this is? It's actually some cells. These things are cells in the lens. So this thing is, it's just hard to believe that this could actually be biology, but this thing right here, this little brick, is a cell. So this is a single cell in your lens, and they've got this great little, check out this down here. They're all sort of like got these little mortise and tenon joints that sort of stick everything together to make the lens really sort of bouncy. So that's actually the lens, if you can believe it. Starts off, remember, as just a little in-pocketing. It's just like this little in-pocketing. It forms a little vesicle, but it eventually turns into this picture. Okay, so last but not least, let's actually look at the geniculate. So this is, that's a picture. Well, we'll just do this very quickly. So this is a picture of the retina, actually. So down here is the pigment epithelium. And then these guys are the little photoreceptors. So I drew them, with a bunch of little folds in them. That's the actual photoreceptor touching against the pigment epithelium. And then these are some of the cells of the photoreceptor cell bodies. And then you've got several different layers as you come through the retina. This is the top of the retina over here like that. So that's the surface of the retina. So the light's coming through all that junk, trying to hit the photoreceptors down here. Okay, that's just a picture of one close-up photoreceptor, so you can sort of see so you can see all these different little layers. That's one of the little in-pocketings in the photoreceptor. Okay, and last but not least, let's look at the geniculate. Okay, so finally, here's what we've been talking about today, which is the geniculate. So where is geniculate? So it's down here. So you can see it's actually, it sort of looks like what I drew, sort of upside down taco. And this is the optic tract coming into the geniculate right there. And here's the rest of the thalamus. If you remember my very basic thalamus lecture, there was the epithalamus, that was a little piece like this. And then there was the dorsal thalamus. This sort of came around like that. And then there's the ventral thalamus, which kind of wraps around. This guy is part of the ventral thalamus like that. And it turns out the geniculate, the dorsal geniculate, it's part of the dorsal thalamus, but the reason it's called a geniculate is because it's like a little sort of knee, because it sort of rotates off the main part of the dorsal thalamus. So this is the dorsal thalamus. And this guy down here, you're getting down into the hypothalamus down here. This section is not exactly perpendicular to the neural tube, so you can see you've gotten down into the midbrain here, the red nucleus. That's another one that we talked about. And here's the substantia nigra down there. It's another one that we sort of mentioned in the midbrain. Okay, so that was kind of like a faraway view, but if we sort of zoom in, now we can actually see those layers that I was talking about. Remember, they started at the bottom. So if we look, so here's one of those coniocellular layers. You can see they're not unobvious at all. There's a bunch of little cells in there. It's just like people said, yeah, we don't care about those. So you can also see the difference between contralateral and ipsilateral. So you can see how this layer comes and kind of stops, and then this layer sort of continues. So that's an example of where one layer is representing, there's no equivalent of this layer here. So this part is the monocular part, this part right here. So it's only seen by one eye. But this whole thing that we're looking at here, these are the larger cells that are the Y cells in layer 1 and 2. So those are the ones that are more sensitive to the change in the signal rather than the amount of the signal. And then these are the smaller cells, like the parvocellular cells, that are more sensitive to the amount of the signal as opposed to the change. But remember, both of them are sensitive to both. So these guys sense change in the signal, but they also sense the amount to some extent. These guys sense the amount of the signal, but they also sense change to some extent, just less so. Okay, sorry I went so far over, but you can get all these pictures in the PDF.